Okay, hello everyone. Now, welcome to the module 4.4. This module will talk about ethics, research integrity, and data sharing. It will be very interesting module, I hope. Now, I will start without wasting so much time. Now, why ethics matters in bioinformatics? Okay, you will say, sir, we have to just gather data, we have to just do this thing, some clicks, and then we will get the results. No, it's not like that. Why ethics matters? See, bioinformatics deals with the increasingly large and valuable datasets. So daily number of datasets are getting added in the databases. So they may include human genomic information, clinical information, population level data, patient derived molecular data, sensitive metadata, etc. So computational capability must be accompanied by responsible scientific practice. Now, what is the research integrity? Research integrity means conducting research honestly, transparently, responsibly, and according to accepted scientific standards. It's not like I'm doing that's a research. Okay, we have to follow some standards. We have to follow some principles. So, important principle includes honesty, accuracy, transparency, reproducibility, proper attribution, responsible reporting, appropriate data management. Now, fabrication, falsification, and plagiarism. What are these three things? Now, these are the research misconducts. Okay, bad practice in the research. I will tell. Fabrication. Fabrication means inventing data or results that never existed. Okay, we in this researcher invent some data or results that are not realistic. Falsification, manipulating research materials, data, processes, or results so that the research record is misrepresented. Means here we manipulate the data. Plagiarism. Here we do the use the another person's ideas, words, or work without appropriate attribution. Now these principles apply to computational research as well as laboratory research. So both the things we do this thing. Now example of computational research misconduct. What generally people do in the misconduct. Suppose you obtain the 100 significant genes, but you reports only 10 genes. Why? Because those 10 support the preferred hypothesis. Okay. So without transparently explaining the selection, this can create a misleading interpretation. So good practices report analysis criteria, filtering criteria, statistical thresholds, exclusion criteria, relevant limitations. Okay, you just get clear from your side. I got 100 results. I got 100 genes. I have used this criteria, this filters, and I got these 10 genes are supporting my uh, hypothesis. Okay, so that's it. So this is the good thing. But if you are doing this thing, you are hiding something or you are not mentioning something. So this is the misconduct in the bioinformatics. Now, reproducibility. A research analysis should ideally be reproducible by another researcher. For example, um, you have performed any experiment in the lab. Okay, so you have to follow one manual. You have to follow one handbook. So if you follow that handbook, you get the results as per the expected results mentioned in that handbook. This why this is so. This is called as a reproducibility. So uh, in the bioinformatics, reproducibility can involve documenting data set or version. You have to mention these things. Okay, data set, version, software version, parameters, reference databases, analysis workflow, statistical methods, code, scripts. So principle is if the analysis cannot be reconstructed, independent verification becomes difficult. Okay, so your result should be verifiable. So that is called as a reproducibility. Data sharing. So here scientific data sharing allows other researchers to verify findings, reuse data sets, conduct new analysis. compare results, build upon previous research. So if you share your data, then your findings will be verified, or anyone can reuse dataset to produce their own analysis or produce their own test or something. However, data sharing must be balanced with privacy, consent, legal requirements, and appropriate access controls. Now, here, important thing, open data doesn't mean everything is public. Okay? Particularly for human biological data, unrestricted public release may create risk. Okay, so what are the potential concerns? Privacy, re-identification, consent limitations, sensitive clinical information, misuse of data. So to avoid that, you just know about what are the things, what is important, what should be hide, what should not. So these things are important here. Now, data attribution. When using another researcher's data, appropriate practice may include citing the original publication. So you have to cite original publication, or citing the database resource where you have, from where you have downloaded or from where you have collected this data, following the datasets usage conditions, acknowledging data producers, respecting licensing requirements. So important principle is data availability does not eliminate the responsibility to provide attribution. Okay, so you have to attribute. Now, fair data principles. What includes in this fair? Fair, F means findable. Data should be discoverable. A. A means accessible. Data should be accessible under appropriate conditions. I for interoperable. Data should work with other systems and standards. R means reusable. Data should be sufficiently documented for meaningful reuse. Now, responsible interpretation. Consider a computational result. Gene X is predicted to be associated with disease Y. Now, this doesn't necessarily mean gene X causes disease Y. Okay, see the difference or see the thing between the words. X is predicted to be associated with the Y. We are not saying that X causes disease Y. So this evidence may indicate, see, association may indicate association, correlation, prediction, functional similarity, something rather than causation. So researcher must distinguish evidence, interpretation, hypothesis, and validation. So this thing you have to see about the things. So for entire theory part of this MOOC, that's it for the entire course. Thank you from my side. If you have any doubt, you just get connected with me using the Google form or using the WhatsApp, or you can directly call to me. Please give like, share, and subscribe to this channel, and stay with me for further announcements and for further workshops and courses. Till then, thank you, and we'll meet in the demonstration session. Thank you.